Ooh. And then, after I wanted to talk about potatoes, I thought, I read this. One of your submissions for the extra credit assignment talked about this, and it really inspired me. I was reading this yesterday, and my comments for class today were all set, and then I thought, you know what, we could talk about epigenetics next time, because it has everything to do with genetics. A little bit less so the traditional breeding of the potato varieties. So what is this? Seedless watermelon. Another GMO. Delicious GMO. So how do you make a seedless fruit? Does anybody know? You could look at this if you want to, but...
The seeds are important, so they never develop.
Right, so you need to find some way to produce plants where the seeds are sterile. Basically, they never become functional, they never develop into actual germinatable seeds. So you can't fertilize pollen, ovum. This is how, thank you very much to the one of you that sent in this from the extra credit assignment. Put me onto this. The goal here is to create a triploid organism. And triploid organisms are sterile because they've got an odd number of chromosomes. So when you go through meiosis, as we talked about early in the semester, you can't evenly sort an odd number of chromosomes into 4 gametes. And so normally, plants that have an odd number of chromosomes, or animals for that matter, would be sterile. So all of the watermelon plants you eat, if they're seedless, are triploids. And that's why they don't have seeds. The seeds didn't develop. They're there, they're just tiny, and that's because they didn't develop to become mature seeds. So then the question is, how do you get a triploid? So you can't propagate, well, you can in plants. You can take cuttings and so forth. But since you can't sexually propagate a triploid, how do you produce an organism that has an odd number of chromosomes? You take a tetraploid that's got 4 copies of every chromosome and a diploid that's got 2 copies of every chromosome. So when those gametes fuse, I know plants do not have flagella on their pollen, but that's okay. What happens when you add the 4 chromosomes from a tetraploid to two chromosomes from a diploid? You get 6 copies, or 3 pairs. So you've made a triploid. So that's exactly what watermelon farmers do. They take a tetraploid watermelon and a normal diploid watermelon, they breed them together, you get a triploid offspring. And then it produces fruits that are sterile, except that in watermelon you have to have fertilization to occur to make fruits develop. So even though you can't get offspring from a triploid plant, they have to have some normal, that's what this is, pollen from a diploid. You have to keep, if you're a watermelon farmer, some normal diploid plants outgrowing in your field, because you need that pollen produced by those normal plants to land on the flowers of the triploid plants to stimulate them to produce fruit. Otherwise, you have triploid plants that don't produce fruit, which is kind of a waste. And that's what that stenospermocarpy means. It's the term for that process, where pollen has to land on the flower to stimulate fruit development. So why the hell am I bothering to tell you all this? How did they make a tetraploid watermelon? What's on that arrow there? What's that chemical? Colchicine. Colchicine. So the farmers chemically treat, well, not necessarily the farmers, the people that produce the tetraploid watermelons treat normal diploid watermelons with a toxic chemical that makes them become tetraploid. So colchicine is highly toxic. It's produced by some crocus plants, actually. It's also an effective anti-tumor agent and does some beneficial things. It's medicinal, but it's toxic. So how does that make you feel about seedless watermelons? They're genetically modified they're delicious. They're genetically modified This is kind of traditional breeding, except for how we got the tetraploid plants in the first place was it involved a toxic chemical. Does that make them less safe to eat? Right, the chemicals, that's the important point. That's, if you take anything out of this, that's the point. Is that just because a toxic chemical happened to produce this tetraploid plant, that was a generation before the triploid plant that produced the fruits that we eat. So it's well removed from our mouth, basically. So do the chemical treatment part in the laboratory, eat the delicious fruit afterwards. So you'll probably get more of this if you take Biology 103, or if you have been taking Biology 103, what colchicine does. It destroys microtubules. It prevents microtubules from forming. Why do I bother mentioning this? What do microtubules do with regard to meiosis? Pardon? Yeah, they pull the chromosomes apart. So what happens if you throw colchicine on a plant and it's trying to do meiosis? How does this figure change? This bit right here doesn't happen. If you don't have microtubules to separate the chromosomes into two separate cells, guess where the chromosomes go? They all, exactly right, they all go in the same cell. So after the first division, you get the 4 sister chromatids, the tetrad stays in one cell. And then the colchicine is still around. So what happens after the second division? That's division 1. So what happens when we do the second meiotic division? Each of those divides. It's like the shell game. Where do you find the chromosomes? They again don't segregate properly. They stay together in one cell. So in this way you make a diploid, or rather a tetraploid, gamete. So all it takes then is fertilization of that tetraploid gamete with one of the gametes like that. So you put one of the, say a pollen grain, for example, that has no chromosomes. Because of the same process, colchicine produces some gametes that have 0 chromosomes and some gametes that have 4 chromosomes. So you do that fertilization, you produce a plant that's tetraploid.